Okay, everybody, welcome to yet another episode of WBESS, Western Biology Evolution Seminar Series. I am your host, Matheus Sanita, and today I have the pleasure to have Gustavo Barroso as my guest. Gustavo is a postdoc at the University of Wisconsin-Madison in the US, and he will be talking to us about his recent work during his postdoc. So, Gustavo, thank you again for being here with me today, and take it away.
Yeah, thanks. My pleasure to be here. Thanks for the invitation. I'm gonna talk about something that I've been kind of obsessed with since the end of my PhD, which is disentangling patterns of variation. And specifically in this talk, we're gonna focus on revisiting the roles of mutation recombination and selection. So when we talk about patterns of variation along the genome, this is what the landscape of diversity may look like. These are nucleotide diversity, average pairwise nucleotide diversity in the Yoruba population in humans from the 1000 Genomes Project, computed in 100 kb windows along chromosome 22. And here we are only including windows that have at least 25% coverage in each of them. That's why there is some discontinuity. But what we see here is that average pairwise diversity, pi, varies substantially along, for example, in this case, human chromosome 22. Between minimum and maximum, that represents nearly a 50-fold difference. And the main goal of this talk is, well, start explaining what shapes these fluctuations in diversity along the human genome. Why do we care? Lewontin, in his classic treatment of evolutionary genetics, his 74 book, he claimed that a description of this genetic variation in a given population is actually the fundamentum datum of evolutionary studies. We need to explain the origin and maintenance of this variation and to predict its evolutionary consequences. That's what we were trying to do here today. So the story begins, or with a landmark paper by Begun and Aquadro in 1992 that proposed that link selection shaped patterns of diversity in Drosophila. What they found was, with the first available DNA sequences and recombination map of the fruit fly Drosophila, they found a strong positive correlation between pi, our measure of nucleotide diversity, and recombination. Here, coefficient of exchange is just another name for the measure of recombination. And this positive correlation was explained by them in terms of link selection. And at the time, 1992, the available model of link selection was genetic hitchhiking driven by selective sweeps. That is to say, link selection caused by positive selection in some parts of the genome. So this work motivated the development of background selection theory, which is the counterpart, you can say, of hitchhiking, because it's link selection caused by negative selection in different parts of the genome, as well as motivated studies of the same kind in humans. Sure enough, when we look at our own data, we see a very strong and positive correlation between recombination rate and pi, as Began and Aquadro did for Drosophila. And here, instead of looking at different genes, we are looking genome-wide. These are the same 100,000 base pair windows as we saw in our first slide. Well, the model of link selection predicts that recombination increases diversity because essentially it decorrelates evolutionary histories of sites in the same chromosome or nearby sites. That is, neutral sites, when recombination rate is high, they evolve less dependently of surrounding constrained sites, and they can, recombination kind of free ups their evolutionary histories, so they can, like variants can segregate at higher frequencies. And so the model of link selection, background selection, as well as hitchhiking also predicts that diversity correlates negatively with the density of constrained sites. That is, sites in the genome, in different parts of the genome, that are the actual targets of direct selection. And once again, looking at our data, trying to explain the landscape of diversity in the first slide, this is chromosome 22, we see that diversity correlates negatively with this density of constrained sites. However, we choose to define these targets of selection. It could be genes, exons, promoters, enhancers, or some other kind of functional elements like CAT scores or FASTCons that we are going to talk about later. And so both of these features, recombination and the density of constrained sites, predict diversity and is compatible with models of background selection. So in this talk, first, we're going to review some efforts to explain human diversity by virtue of background selection. We're going to introduce our new model of BGS, which we call MOMES++, and is just about to be published in Genetics. And we're going to finally embed these predictions that we generate with our population genetics model together with publicly available genomic maps in humans, because in humans now, of course, it is our species of strongest interest. We have high resolution maps of recombination, mutation, and selection. That is to say, we have a pretty good idea of different sites in the genome, how often they're recombining, how often they're experiencing mutation, where selection is happening, and how strongly. And we're going to take all of that and put that into a causal inference framework to drive to try to explain diversity better. So B values, where the B stands for background selection, they are the classic statistic that measure this relative diversity in a focal site, let's say a focal neutral site, which we can call in this case locus A, as Hudson and Kaplan did in 95 when they introduced their model. And we're trying to model with population genetics theory how diversity at our focal neutral site is reduced by virtue of selection happening around it. And what this means is because there are genes that are linked to our focal neutral site A, there is a somewhat constant influx of deleterious variants just by the process of mutation, and also a selective pressure that is a removal of these deleterious variants at linked sites by negative selection. And so as a result of this, diversity in locus A will tend to be dragged down alongside with diversity at the other loci, in this case, 1, 2, 3, 4, all the way to M, that are the subject of direct selection. And so this was one of these classic papers that introduced background selection with recombination in the 90s in a series of papers by Charlesworth as well and others. And well, they used Drosophila to compare predictions with the data. And so the model here on the left, we just have a little the analytic prediction of what diversity should be or is expected to be at each focal neutral site, and it's going to be a product of the effect of selection happening on the other M loci that are linked to it, right? So it's going to be a function of this pi naught, which is what diversity is expected to be if there was no link selection at all. And then we're going to multiply together the individual contributions of link selection from the other sites in the chromosome. And so this, you don't need to care like about the functional form of this, is not the point. I just want to call your attention that what matters here is mu of xi, that is the deleterious mutation rate in each of these functional elements, sh, which is the coefficient of selection acting against deleterious mutations in these functional elements, as well as this R, big R here, which stands for the recombination distance between our focal neutral site and the various constrained sites in the chromosome. So with this, we need the individual mutation rates, the recombination map, and selection coefficients, and these are all the ingredients that we need to make predictions about what we expect diversity to be. So if we have all of these ingredients, then we can start to make predictions, as they did in 95 with the Drosophila data here, with a handful of genes uh, whose diversity is observed here by these big circles with crosses. This is the observed data. And we can see that the predictions match the data reasonably well, already with this very simplistic model and data from 95. And we are trying here, they were trying with different selective pressures against the deleterious mutations from 3% all the way to like down to 0.5% of a decreasing fitness for each deleterious variant. But we see that there is a pretty good match between predictions and observations back in 95. So we move forward all the way to 2002, when Murphy and collaborators fit these background selection models using whole genome sequences in humans for the first time. Again, they were using here something like 1,000 genomes data, and they had the ingredients that we saw were necessary. They had the position of the constrained sites. Here they use CAT scores, the recombination map, but they assume a flat mutation rate. Well, using this same model that Hudson and Kaplan did, they predicted that this classic BGS can explain around 60% of the variation in diversity in the human genome in 1 megabase windows genome-wide. They also found that sweeps or genetic hitchhiking, that is link selection caused by positive selection, has a negligible effect in humans, as others had suggested already. Only 2 years later, Buffalo and Kern, instead of using the classic background selection model, they used another model developed by Santiago and Caballero, which besides working in the strong selection regime, the model also works for weak selection. And they saw that by including the effect of weak selection, you can modestly improve the R squared figure here, the amount of variance that are explained with this model, to around 67%. Again, when you look at the megabase scale, that is looking at 1 megabase windows across the genome. Now, they went further and they suggested that, in fact, 67% is as much as you can possibly explain in the variation in diversity in the human genome, because they argued that drift imposes an upper limit. That is because genealogical noise, stochastic fluctuations in the coalescence along the genome are going to account for at least 33% of the variation in diversity. And by explaining 67%, they claim that background selection explains all the diversity that can possibly be explained in humans. Well, we can ask, what about other sources of genetic variation? For example, around 10 years ago, people started looking at mutation rate variation in humans. It took a long time for us to start digging into this in this mutation landscape, in the so-called mutation maps, because they are notoriously difficult to estimate. But you can imagine that if the mutation rate changes in different parts of the genome, that's also going to affect levels of genetic diversity. And when people started doing that, they found that features of genetic diversity like allele frequencies and site frequency spectra, they are also heavily explained just by mutation rate variation. And sure enough, when we look at our own data, using the mutation map that is available to us now, that is the roulette map that was published by Cipliarski and others in 2023, we see that the mutation landscape is as well strongly correlated with genetic diversity in our favorite chromosome 22 that is shown in the first slide. So what's going on here? There's been a traditional focus in explaining genetic diversity along the genome, which is an obsession of population geneticists. There's been a traditional focus of one evolutionary process at a time. That is, either you focus on link selection and you pretend mutation rate is constant, and you see how well your model does, or you focus exclusively on mutation rate variation, pretend there is no link selection, and see how that model does. But the problem is that without something that puts this together, these correlation-based studies are descriptive and they have basically talked past each other. What we want here is to build more realistic models of human diversity. Why is that? Well, first we need a better model of background selection because we believe that they likely overfitted how much variance in diversity can be explained by these models. In particular, even the Buffalo and Kern paper or model, they use the Santiago and Caballero model, which is already better than classic background selection because it incorporates the weak selection regime, which you see here to the right of this plot. You see that there is a discontinuity in the predictions. That is, the model cannot predict B values for selection coefficients around -1, scale selection coefficients, that is, NS of around -1 here. And that's a problem. And that's a function of the mutation rate as well. And human mutation rates on average are 1.25 times 10 to the minus 8. So that is higher than the red curve that they propose to benchmark here. There is a problem will be higher using the human average mutation rate, and even more so because we know that the mutation rate fluctuates along the genome. So this discontinuity may have created a problem in their predictions. And also, and perhaps more importantly, the way that they arrive at the R squared of around 67% is by fitting selection parameters based on the landscape of diversity. That is, once you have the targets of selection, here, for example, in the genome, that is fixed, but they fit the deleterious mutation rate as well as the strength of selection acting against deleterious variants that are happening in different parts of the genome, in different genomic elements, that say exons and fascons and all other kinds of genome annotations. These parameters are being fitted in an iterative procedure that minimizes the distance between observed diversity in blue and that predicted by the model. And then essentially by doing this, they are choosing the parameter values that by definition maximize R squared. So they may have overfitted this. In contrast, our model does not have the discontinuity in prediction in NS, as you can see here in an analogous plot to the ones that I showed you before. For the different mutation rates, we can predict B values accurately. Simulations here are shown as the circles, and then the predictions are shown as the lines. You see here that this benchmarking shows that we can predict mutation rates accurately across the full distribution of fitness effects of mutations, especially if we incorporate interference, but that's we're not going to get into detail of that, but we do incorporate interference, and then our naive predictions were not so good in the solid line, they become much better with the dashed line here. So this is an important improvement. And then we benchmark thoroughly by simulating 1,000 replicates of human chromosome 2. And here we have, how do we simulate that? We do forward simulations, forward pi 11, using some recombination map that we show at the top, some distribution of exons here shown as the purple circles, and a distribution of selection coefficients that is saying how strong is selection against mutations that happen in these different exons. And so the blue dots here are average B values over these 1,000 simulations. And we see that our model does a remarkable job at getting the B values correctly. For example, here looking at this region of 10 megabases of the human chromosome 2. What this means is if background selection truly is the only mode of selection that is happening in the human genome, then we'll be able to infer it quite well. And sure enough, well, now we turn to data, real data, and the b-values are again strongly correlated with genetic diversity in these 100,000 base pair windows of human chromosome 22. So what we observe is we have different maps that all correlate with diversity. We have the recombination map, the mutation map, the map E here that we call the functional elements along the genome, and the B-value map, and they all correlate positively with diversity, or that is, the square Pearson correlation is high across the human genome. What I'm showing you here in the triangles are correlations in individual human chromosomes at the megabase scale. The black stars represent the genome-wide square correlations. And we see, for example, mutation rates, genome-wide is about the same as the square correlation with diversity. Is a bit lower here for the distribution of functional size. But here, let's focus on the B values, which are the target actually of these population genetic models. We have the buffalo and kern standing at 67%, Murphy and red at 60%. We have our distribution of square Pearson correlations across the chromosome, which is fairly wide and something that we documented for the first time because previous studies focused on genome-wide correlations alone. But when we look at our genome-wide correlation in the black star, it stands around 50% only. And I'm going to recall that our model is more accurate, number one. And number two, we don't do the model fitting procedure that estimates selection parameters that creates some kind of circularity in the predictions by maximizing our squared. Instead, we use estimates from the literature. We used DFEs based on the size frequency spectrum that other people have published. And we use mutation and recombination maps, and we fix those when predicting b-values. In that way, our model is more parsimonious. So the conclusion here is because our improved model correlates less strongly with diversity, is that background selection does not track it as closely as previously thought, because those methods were likely overfitted. So you can probably guess where we're going with this. There are a lot of things that are correlated with diversity. But these individual R-squared values, they cannot be interpreted as measures of relative importance because they conflate paths of influence. That is, B-values, functional elements, mutation rates, and recombination, they do not affect diversity directly themselves, but instead they affect each other, and sometimes indirectly or directly they affect diversity. But looking at this individually and trying to interpret this as partition variance explained wouldn't work because, for example, if you look at the different scales here, focusing at 1 megabase scale, which is shown at the right, if you would treat them independently and sum their individual contributions, well, you would explain more than 100% of diversity, which just exposes the logical fallacy of doing so, of analyzing correlations individually and independently. We need a method to look at these processes jointly and then trying to estimate their relative importance in shaping the landscape of diversity. So we want models in the end, they are not only predictive, but also explanatory. So using our method, this is what it predicts for the same landscape of diversity that is used to start this talk, right? So we need to define the targets of selection. These are, we use two types of functional annotations. In red, we have predictions as if the targets of direct selection were genes. And by genes here I include regulatory regions, promoters and enhancers, and exons. And again, I'm using DFEs that have been inferred by other methods and in other papers. This way, I'm trying to remain parsimonious. And you see that when you fit this model, or when you use these parameters, and on top of that, we use the roulette mutation map, it does a pretty good job at predicting the diversity landscape shown in black. the observed one. But a different method, a different set of annotations, which is known as FastCon scores. FastCon scores are a computational method to predict where are the targets of selection across the genome using a phylogenetic, like using a vertebrate phylogeny and finding regions that are highly conserved across 99 vertebrates. And if we use this definition and GFEs that have been inferred from the SFS, you see that the FastCons predict diversity much better than the genes model as well. But how do we move forward from here? Well, we can make nice predictions, but how do we go from prediction to explanation? Well, the core issue in the end is that we all know that correlation is not causation, of course, but it does imply a hidden causal structure that perhaps we can learn from the data. Here, just a toy example. There is a very clear trend of Nobel Prizes normalized per population and chocolate consumption. That is, the more chocolate you eat, the more likely you are to win a Nobel Prize. But of course, this is not a direct cause. But these two variables are linked because there is some underlying causal structure that perhaps we can learn. And here, this is actually a subject of study. This author attempted to explain this, and the conclusion is it remains unclear whether the correlation is spurious or an indication for hidden variables. Okay, how do we get there? But first, there is one question that I must address, and it's not trivial to do so. Is the pursuit of causal explanations an exercise in futility? And that is, I spent quite some time in saying that it's important to explain the landscape of diversity rather than just predict it and maximize R squared, maximize correlations. However, population genetics theory already provides us with models, mechanistic models, detailed models, that describe how drift, mutation, recombination, and selection interact to shape expected diversity. And in fact, our method, Moments++, that predicts the b-values that I showed before, it is based on precisely this body of theory. So am I working too hard here in trying to explain diversity? Well, if mutation, recombination, selection interacted only through background selection, then I will agree with a skeptical viewer that would say this is an exercise in futility because untangling their contributions to diversity will be straightforward in this case. But the issue is precisely that they can affect diversity both directly and indirectly. And so we can view this as understanding diversity requiring an integration of other disciplines as well, in particular molecular biology. So how do we move from correlations to causation then? Well, already 100 years ago, Sewell Wright, notorious population geneticist, he worked on his path analysis method that attempted to do precisely that. He says, the ideal method of science is the study of the direct influence of one condition on another in experiments in which all other possible causes of variation are eliminated. And unfortunately, causes of variation often seem to be beyond control, as we all know. And the degree of correlation between two variables can be calculated with well-known methods, but it only gives the resultant of all connecting paths of influence. So this is the proposed path analysis or the path model, structural equation model that I present to you today to explain the landscape of diversity. What do we have here? How do we predict diversity? Here we have a couple of ingredients that are available in humans. For example, we need a recombination map R, the mutation map, mu, the position of constraining elements that we call E, diversity is the observed one that we have in the Yoruba population, for example. And using E, mu, and R in our method for background selection, we can predict B. So Moment++ is going to give us B predictions in different windows of the genome. And then these directed acyclic graphs that I'm showing you, they are attempts to specify how these variables affect each other causally. Each arrow here, a path coefficient to be estimated by the method, says that, for example, mutation rate is going to affect B values directly because B values depend on the literature diversity, and also has a different path to diversity more directly. That is simply because regions under a higher mutation rate will tend to be more diverse, other things being equal. Also, we have a path from recombination to B values because we know that, as has been studied since 92 and in the Bagan and Quadro paper, regions of high recombination tend to have decoupled evolutionary histories and they tend to increase B values, therefore increasing diversity. But also there is another path here from recombination to mutation rate because in humans, there is a strong amount of evidence suggesting that recombination is mutagenic. And so these path models here, causal models, are a way to explain the relationship among the relevant variants beyond their simple effect on pi. And we can extend this model with a subtle but important covariance term here between the density of constraint sites and the local mutation rates. This is a topic of debate in the population genetics and evolutionary community here, whether mutation rates are lower or higher in different constraint elements along the genome. But the point is that, so now we have two candidate models that propose to explain these variables, and by modeling the relationship or the connections between these variables beyond their effect on pi, we can explain diversity better. So how do these structural cohesion models, or SIM, how do they work? Well, in a nutshell, you have a set of variables, and this set of variables is going to give you an observed covariance matrix, some description of how these variables are correlated with each other. And we are going to fit these path coefficients in such a way that minimizes the distance between the observed covariance matrix and that which is predicted by the model. And so what are the advantages of this? Well, we can model hierarchical relationships and not simply isolated predictors, and therefore we can separate direct and indirect causal effects. We can estimate multiple link regressions simultaneously. Sometimes, and this is something that we do here, but I'm not going to go in detail, we can use latent variables, that are variables that are not directly observed in the data, to capture underlying constructs and reduce measurement error. For example, we have an available mutation map for our humans. We have more than one now, in fact. But these are not necessarily the true underlying mutation map because they carry some error with them. And SIM allows us to take that error in the individual estimates into account and represent this underlying construct that is supposed to be the true mutation map more faithfully than the individual estimates that we get from the literature. Finally, and this is something that I value a lot in this kind of methods, is we can test whether a proposed causal structure matches the observed covariances. That is, we can test the causal structure that we propose in our model, which is a big advantage of SEM over traditional regression. So moving on to the actual data, we have is that the proposal models fit the data well in most chromosomes. So I didn't show you here, but the first result that we have is that fitting a genome-wide model is offers a poor fit to the data in comparison to fitting chromosome-specific models. That is to say that the effects, the relative contributions of background selection, recombination, mutation to diversity, they vary by human chromosome. This is something that is a level of variation that was previously undetected. But in terms of overall fit, we can compute these fit indices, let's say the comparative fit index, CFI, or goodness of fit index. And here I'm showing you the fit indices for the different human autosomes for the model 3 in red, or the model 4 with the extra covariance term in blue. Um, And in general, values above 0.9 are indicative of an acceptable or good fit. And for most chromosomes, especially for the DAG4, which includes the covariance term, the model seems to be doing a good job at explaining the observed data. For example, here, maybe not so much for like a couple of chromosomes, but for all the others, the fit indices are reasonably good. So what does that mean? Well, when we look at the actual parameter inference, the values of the path coefficients that we have from one variable to another, and we standardize these variables prior to fitting the model, this means that the coefficient, like these variables, they don't have any units, and the coefficients directly give an estimate of the relative importance. The observed pattern here from these results is that the path from B, the direct path from B to pi, tends to be higher than the direct path from mu to pi. Here I'm just showing you the outlier here that predicts a negative effect of mutation on pi. This is chromosome 19. Chromosome 19 is one where the model didn't fit well, and that I showed in the previous slide, one of the two under chromosome under DAG4. And that's like chromosome 19 is a problem, is a known outlier in according to several features, exonic content, GC content. And so in this particular chromosome, the model clearly failed. But in others, we see that the effect of the background selection is stronger than the effect of mutation rate variation in shaping diversity. But the effect of mutation is never trivial. Is sometimes in some chromosomes is on the same order here. The effect of mutation is comparable to the effect of background selection. Another thing that we see, and here I'm only showing you, like, for example, here we don't have 22 points. The reason for that is I'm only showing you path coefficients that are estimated to be statistically significant. I'm showing you here that the indirect path from recombination to mutation to diversity tends to be lower than recombination to background selection to diversity. This was a debate in human population genetics because we know for a long time that recombination affects diversity via these two different pathways. But here we can show, we can estimate at least for the first time their relative contributions, and we can propose an explanation to this debate, suggesting that the effect of recombination via background selection by alleviating the effect of background selection on diversity is higher than the mutagenic effect. We also see large variation in R squared of pi among these autosomes. Structural equation models also gives us this measure of variance explained in diversity. And we see that in the overall model, once we account for recombination mutation as well, and density of constrained sites, we tend to explain actually more diversity than in some chromosomes, at least, more diversity than in previous methods, because we are incorporating this molecular biology component. But R squared of pi varies from just under 60% in chromosome 6 and 14 to 92% in chromosome 22. This is a large amount of variation here. So we realize that chromosomes that stand at the top or to the right of this wide distribution R squared, what they suggest is that once we incorporate molecular biology, we can actually predict and explain diversity better going beyond the 67% limit that was suggested by, by Buffalo and Kern. So we can exceed that limit. So drift does not actually impose at least 33% of noise. Our model suggests that we can do better than that. And the fact that we have such high variation, and the fact that in some chromosomes we do a better job, this motivates the search for what the reasons, the biological reasons that may be driving this among chromosome differences. So we know that chromosomes vary in several features, several genomic features, such as average mutation rate, average recombination rate, the density of constrained sites. I mentioned that chromosome 19 is an outlier, is the one that has, for example, the more genes, the more exons and genes in the human genome. They also can vary in GC content. Well, we postulated that this large variation in R squared could be to some extent driven by these different average features that chromosomes may have. And to sort this out, or in a descriptive way, we resorted to linear regression, where here we're trying to explain the R squared of pi of a given chromosome. So we have 22 points. Each has its R squared of pi predicted from SEM as a function of the linear contributions of their average mutation rate, recombination, density of constrained sites, and GC content. And in the end, across all scales, we see that the only such feature with a significant and positive effect on diversity is GC content. That is to say, we explain diversity better in general. Looking at these 22 human autosomes, we tend to explain their diversity better where GC content is highest. And this is going to be especially true if the targets of selection that we focus on in the background selection model are genes, exon promoters, and enhancers. To some extent, this is going to be true for FASTcons as well, but not so much. And especially true for FASTcons at the 10 kb, but this effect gets diluted at 100 kb and especially at the 1 megabase scale. So what are the take-home messages here? Well, we find that indeed, once we consider all things in our causal model, we propose a causal model, we don't consider all things ever, but is a very comprehensive, these are very comprehensive models. Once we include these variables and their relationships, background selection stands out as the most important evolutionary force shaping human diversity, but not as strongly as previously believed. And in many autosomes, mutation rate variation is a very meaningful effect. Recombination impacts diversity more strongly through its background selection effect than by mutagenic effects. And this helps to resolve a long-standing debate that goes back to the early 2000s. And we have that explanatory power varies widely across chromosomes, which is a previously unrecognized heterogeneity. So just to finish, let's take a step back. And I'm showing you like visually in terms of the landscapes and observed data and predictions. I have focused on chromosome 22. And chromosome 22 is the one where we explain the most variance in diversity using our models. But what about other chromosomes that don't look so good? For example, here we have chromosome 13. We have the observed landscape of diversity in black in 1 megabase windows now. And we have the predictions using the background selection model and the mutation map in the colors as we had before. So for red, we have the predictions under the genes model as the targets of selection. And in green, we have the predictions under the FASTcons model. But we see that this does not look nearly as good, even at the 1 megabase scale, where we have less noise than the 100 kb scale that I've shown you so far for 22. This does not look nearly as good. So in some areas, indeed, we get it right. We get the trained correctly, and it's fine. It doesn't do a terrible job for sure, but there is a difference between our best chromosome and this one. This is chromosome 13, one of the worst. And so what could be causing is something that we don't have a definitive answer, but we can try to speculate a little bit. There is no obvious a priori reason why some chromosomes should fit better than others. There is nothing obvious about evolutionary biology or population genetics that says that the effect of selection mutation should fit the diversity better in some chromosomes than others. Um, what about the maps that we use to fit these models, these ingredients that we then not only predict p-values with, but we embed them in these directed acyclic graphs representing causal relationships? Well, these maps are estimated, the recombination map, the mutation map, and in particular, the identification of the direct targets of selection. Uh, we don't have access to the true maps, right? So it could be that in some chromosomes, the maps are better estimated than others. This could be definitely a reason causing this effect. And it could also be that in some chromosomes, background selection is not the only mode of selection that is shaping patterns of diversity, for example. Maybe the purely negatively selected, like the pure model of negative selection or purifying selection, is going to be more adequate in some chromosomes than others. For example, in some chromosomes, uh, stabilizing selection on complex traits, for example, could be generating patterns of diversity that mismatch relative to our expectations of purely deleterious mutations. So this is something that we leave as an exercise for like future studies, trying to understand why some chromosomes fit better than others. But for now, um, we have done considerable progress, I believe, in explaining diversity more than just predicting it. So thank you for your attention, and I'm happy to answer questions.
Thank you, Gustavo. I must confess that in the very end, in your final slide, you kind of stole my thunder because I one of the questions I had for you was like, why is it we have such a difference between chromosomes? Because one thing that I'm really intrigued by, is the evolution of karyotypes, right? Because if you think
Mm-hmm.
that we actually see a good diversity of karyotype organization, so to speak, even within species that are closely related, right? You have, if I'm not mistaken, I think the Cervus genus, the deer family, deer,
Mm-hmm.
different deer species, they have variation like from one or two chromosomes to dozens of chromosomes, and phenotypically they are just the same. So
Yeah.
I've always been very interested in the physical attributes of genomes and looking at them as if they were a anatomic, morphological feature, so to speak. And yeah, and I was that wasn't one of my questions, and then you just said like, look, as of now, there is nothing that really, based on evolutionary biology theory that we know of, that really predicts why we would see such a difference in depending on the chromosome. But you did touch
Yeah.
on a good point in which some certain chromosomes might not be as well mapped as others, right? So there should
Yeah.
be
Yeah. Yeah.
that effect.
Yeah, I especially like functional annotations and also the mutation map, but like particularly in particular functional annotations, like
Mm,
I suspect that in some chromosomes we may simply be like missing a lot
mm,
of where selection could be happening as just not being as neatly as identified as in others. I suspect this could be a big issue.
mhm.
And now more models are like computational models are being published that try to identify reasons of the genome under selection. And maybe, with these models, after we have distribution of fitness effects for the mutations that happens on these elements, we can do better after we have improved models of annotation. Yeah.
Yeah, one thing also related to the chromosome level, so to speak, that I was curious about is that you've shown us several graphs in which they were based on chromosome 22 or chromosome 2, and
Hmm. Hmm.
it stood out to me that those are, the second smallest and the second largest chromosomes.
Hmm.
Is there a reason why you have more data in these chromosomes? Or, and also you mentioned 19 is known for being problematic. Uh,
Hmm.
what's the underlying story, I would say, in that? Like, why always these, these
I,
chromosomes and Yeah, some more context, I would say. I mean, coming from a place of curiosity or
Mm-hmm.
naivety.
No, for sure. I think, okay, first I'm going to give you the one supplemental slide that I have then, since
Okay,
you
good.
touched on it. I will first go there, which is actual bootstrapping. So here I'm not only showing like in the talk I only touched on DAG 3 and 4, which are at the bottom, right?
Mhm,
But the existence of DAG 3 or model 3 and 4 suggests the existence of models 1 and 2, which
mhm,
I didn't touch on, but they're there. But DAG 3 and 4 are more comprehensive, and that's why I focus on them. They're better. They're better descriptions. Touching on chromosome size, here at the 1 megabase scale, I did bootstrapping 5,000 replicates,
mhm,
fitted the model on this resample data, and got the R square of pi. And you see that indeed among the, like, for example, chromosome 22, the smallest one, like there is a lot of variation in R square of pi based on bootstrap, right? So the R square of pi varies a lot depending on resampling. And in other chromosomes, less so. And in general, like you still see a lot of, even though the distributions, when you look at all the data, they overlap, you see that in general, like there is still quite some amount of variation in the variance in the diversity that we explain with these models. For example, like, yeah, it doesn't seem to be just a matter of chromosome size because there is no obvious trend
mhm,
actually in terms of chromosome size. So we have like, sure, we have some fluctuation, but it's not like, oh, clearly in larger chromosomes we explain diversity better, or clearly in smaller chromosomes we explain diversity better. There's nothing obvious stands out. And Bootstrap, besides chromosome 22, suggests that like even though there is variation, especially like okay, chromosome 14, for example, we the model doesn't fit as well. There is a lot of variation, but some chromosomes do seem to fit better than others,
mhm.
and it's not a function of chromosome size alone.
I agree.
And the other thing is that okay, so chromosome 19 being an outlier, right? And is an outlier in terms of well, the constraint elements, GC, but you see, this my causal model doesn't say anything about GC content.
Mhm,
You can ask, well, what about GC bias gene conversion, right? Recombination is not only resolved in crossover,
mhm,
we also have um, gene conversion, which we know that changes the composition of the DNA sequence of the chromosome because it tends to favor GC bonds. And so that's not part of the model. Maybe that's something that should be included in the next model
mhm,
that improves on this one. And maybe we can explain diversity better when we have something like a method for like a causal effect of gene conversions, or we have methylation patterns. Maybe when we have like a methylation map of the human genome
mhm,
that is like is reasonable, some kind of reasonable average across cell types. Maybe once we include that and give that a meaning in this causal framework, we are going to start to do better in chromosomes like 19. And like going to your other question, chromosomes, like the structure of chromosomes, like methylation patterns and other kinds of epigenetic annotations are something that could be affecting the overall covariance structure of the variables. And so by including all, like not all, but other kinds of maps of these chromosomes, maybe we can start explaining the overall structure better.
mhm. Other attributes. Yeah. And it's great
Other
that
attributes, yeah.
you return to this slide because I even made a note of it because I really liked. I found it to be very informative, and I must say that you did a great job explaining, the rationale of your model and linking to what you what you referred as being an obsession within population genetics, which is that
Yeah.
focus on one mechanism only, right? And this is something I think most of us would agree that we often see in the field not of only population genetics, but let's say evolutionary biology. We see folks studying a particular mechanism and becoming sometimes almost dogmatic about it and either loving or
Yeah,
hating that mechanism and trying to explain all the diversity out there based on that one thing, one process.
yeah,
And I
yeah,
think you did a great job in saying, look, we should have a more pluralist or embracing more options when trying to expand diversity. I really like this slide.
yeah,
Very nice.
yeah,
Um,
yeah, yeah.
yeah, you great. I must say you did a really good job explaining, in simple terms, the rationale of your work. So thank you for that. I really appreciate it.
Yeah. Thank you. Thank you. And one thing, well, I kind of stumbled upon
Mm,
this kind of method, the structural equation models, like towards the end of my PhD, because I wanted to explain the landscape of diversity. And so I got a book. The book started saying a few things, and then I got another book, and I started reading those. And this all traces back to Wright in
hmm
1921, which is a population, which he was a population geneticist,
One
one
of
of
the
the
founding
most
fathers. Yeah.
important of all time. But like this, his method of path analysis has eluded population genetics. Like people don't know about this. People instead focus on the correlation method that was developed by Pearson and Fisher. And there was like, of course, there is attention, Wright and Fisher in population genetics, but also in statistical methods, it seems. And in evolutionary biology in general and population genetics, like Fisher definitely won this dogmatic battle or the battle of methods to analyze data with because we don't learn about path analysis. We don't learn about structural equation models. And in fact, I believe that this is what we should be learning about. Like Fisher and Pearson, they had this like positivist view of science
Mm.
where you can only describe but not explain in terms of using causal arguments. And Wright was the opposite. And I think that, well, so social scientists and ecologists were responsible for kind of rediscovering, maybe is a strong word, but continuing the worker right in the 20th century. And they did a lot of work in statistics, the social sciences and ecologists. And and I think there is a lot that we is already available and we can just borrow and start using in population genetics that goes beyond correlation and multiple regression.
Yeah, that's a very good point. I agree with you. I always had that feeling as well, and of um, within, if you think about Fisher, Wright, and Haldane, Fisher usually is the one that comes to mind first. And perhaps one could even make a point of, later, later through the modern synthesis, you know, Gold, I don't think Lewontin would say that, but Gold said that there was the hardening of the modern synthesis in which it became very adaptationist. And perhaps one could even say that from the beginning, there was already this one-sided or one-track mind um, within, within the beginning of evolution in the 20th century. But yeah, really good
Mhm.
point. I also must say, on this note, I must say that I also appreciate you bringing philosophical aspects of how science is done and how our own schools of thought inform our decisions and our
Yeah,
rationale. So this is also really cool. Thank you. I I would
thank
say
you.
if I were to guess, you were a fan of Lewontin in
Yes,
in
yeah,
his
yes,
way of approaching things. And I also really like the picture you have of
yes,
him with Maynard
yeah,
Smith. That was a nice picture.
yeah. No, that's so cool. Like one thing that, like, as I read a bit more about this, and I start, it's nice to learn the history, and
Mm,
because sometimes by learning the history, like some things like the reason why people study
yeah,
a few things start becoming more clear, right?
it makes so
And
much sense.
so
Yeah.
it makes so much more sense. Like
Often,
Lewontin 74, he pointed out like Lewontin's paradox, right? So Lewontin's paradox, so the distribution of average diversity across species is not as wide as you would expect given their population sizes. And then, well, Maynard Smith proposes them in the same year, actually, the model for genetic hitchhiking, which was used by Begun and Aquadro as an explanation for why variation varies along the genome. But the motivation for Maynard Smith and his model of genetic hitchhiking was actually to explain genome-wide average diversity as an explanation, as a potential explanation for Lewontin's paradox, which
Mm,
is something that I had no idea. And of course, they interacted, right? This is a picture from a Christmas party, I believe. I
mm,
think Lewontin was doing a sabbatical with Maynard Smith, and
mm,
so like it starts to click. Why hitchhiking was proposed in 74
mhm,
before we had any idea about the recombination rate, well, any data on recombination rate variation and nucleotide diversity along the genome. Like Maynard Smith proposed a model that was used to explain this, but in fact was used to explain genome-wide average across species diversity, which is kind of crazy.
mm. I agree. I oftentimes learn, well, just like I said, but sometimes I go onto like history of biology journals, and then I actually, when I want to learn more of these side of things, and I oftentimes things make sense when I learn about the history of how my
Yeah,
particular discipline has evolved. So yeah, I agree with you.
yeah, yeah.
Anyway, Gustavo, this was great. Thank you very much for taking the time to explain to us your recent research. You've mentioned at the beginning of your talk that this work should soon be published in genetics. Is that correct?
Oh, sorry. Yeah, that
Or
was
another
a
or
bit of
similar journal?
I conflated things. So let me.
Okay.
This is easy to explain. This is easy to explain. So this method that so this the method moments plus plus that makes b-value predictions
Mhm.
is gonna soon be out in genetics. It's
Okay.
provisionally accepted. I just need to change a few wordings,
Mhm.
and it's gonna be soon be out in genetics. The method itself, but this other like more broad data analysis work that I presented later,
Mhm.
that is something that I'm still working on
Working.
finishing, and we want to publish on bioRxiv as soon as possible.
Okay. Yeah, I was gonna say, uh, do so because like this is really like a hot topic, and I'm pretty sure it's going to attract a lot of attention. So yeah, keep up the good work. This is great.
Thanks. Thanks.
Thank you, Gustavo. So with that, I'm going to stop the recording here right now, and then we can